Hello, everyone. On behalf of the ACS Division of Analytical Chemistry, the Education Committee, I welcome you to this panel. The aim is to motivate faculty to implement cures or course-based undergraduate research experiences into your curriculum. We will focus on the positive impacts of cures on students and faculty and address potential implementation challenges. Please participate in the discussion by typing your questions in the chat. There should be also time at the end for you to ask any questions you may still have. Just please, as a reminder, this session is going to be recorded, so we can then share the recording with a wide audience. And we will also provide the link to a PowerPoint slide. I'm very excited about this session because I do teach analytical chemistry, quantitative and instrumental analysis, as well as general chemistry at Barry University, which is primarily undergraduate institution. And we are looking to expand the authentic research experience opportunities for our students. And cures is one way to do it. And what is the best way to start something new than learning from the experts? And then this brings me to introduction of our panelists, Dr. Ellis Bell, who is the lecturer too of chemistry and biochemistry at the University of San Diego. Dr. Bell has years and years of experience implementing cures into the curriculum. And he's also organizing a session at the upcoming fall ACS meeting. So if you're going to be at the meeting in Denver, block your time on Sunday. Please do attend his session. We also have Dr. Renée, who is a professor of chemistry and department executive officer at the University of Iowa. Dr. Cole is the recipient of the 2023 Norris Award for Outstanding Achievements in Teaching of Chemistry. The shared award is for the collaborative work on the enhancing learning by improving process skills in STEM or Ellis project. She wears multiple hats. One of them is as a previous associate editor of Journal of Chemical Education. And today she will be sharing her advice on designing curricular innovations with publishing in mind. As one of the cited benefits of cures is producing publishable data. And last but not least, Dr. Frederick is a professor of chemistry and Charles Lubin Family Chair for Women in Science at Skidmore College. And Dr. Frederick will introduce us to the new concepts and benefits of implementing microcures specifically into analytical chemistry curriculum. Thank you to all our panelists for dedicating their time to this session, and welcome all of you. From here, our panelists prepared a short presentation and afterwards, introducing us to different definitions of cures and also to successful projects. And there will be a lot of resources. This is why it's going to be so important to also share this PowerPoint presentation. If you, again, have any questions, write them in the chat. And there is also going to be time at the end to ask any questions that you might still have. So with this said, Dr. Cole, the PowerPoint is yours.
All right. Except for you have to change the slide. So as we think about cures, I mean, part of the motivation, if we think about outcomes for chemistry programs and particularly for the laboratory pieces of it, science practices should be kind of what this is all about. And the next few slides, these are things that I have borrowed from PhD student of mine who just graduated last month. So Andrea Van Wyk has done a lot of work with looking at learning in the laboratory and how do we improve science practices and inquiry. So the 9 practices on the slide are part of the next generation science standards, but I've yet to speak to any chemist who doesn't agree. These are all important skills that our students should be able to engage in. They're all embedded in the ACS CPT guidelines for departments. So you'll see a couple of places later where the numbers might pop up, but when we think about designing laboratory programs, you should have these in mind. And where are you giving students opportunities to engage in these practices? And when we think about the laboratory curriculum, you know, you get this range of approaches, and our data very strongly, and everybody else, most faculty, the majority of the lab experiences they offer to students are in this cookbook style that All you have to do is follow the recipe. And we've done work that students will agree with the statement, I can be successful in the lab without understanding what's happening. And if students can agree with that, I would posit that we haven't quite done our job right. And so the goal is to shift students to more learner-directed, and the course-based undergraduate research experiences are on that authentic side. And then you can do some scaffolding type things with inquiry-oriented experiments in the middle. Oops, I lost Susanna. Next slide. And the benefits these have been clearly demonstrated. There's a number of references of research studies there that we see improvements in performance, improved critical thinking, increased self-reflection of learning. It closes performance gaps. I mean, all of the things we want for our students happen when you get students engaged in these authentic laboratory instruction. Often undergraduate research is cited as a high impact practice, but we know that for lots of reasons, not all students can do those. And so cures provide that bridge. Next slide. And this is just here. So Andrea designed a cure in our analytical measurements class on looking at indoor films. And so this was related to active research with one of our faculty. And this is just for a tiny bit of context for the components of a cure, which is the next slide. And so when you think about designing a cure, if you're being consistent with the intent, you should have 5 defining characteristics. So 1, students should engage in science practices. And in the cure Andrea designed, students were tasked with planning and carrying out investigations. So these are the science practices that were highlighted: analyzing and interpreting data, engaging in argument from evidence, and then obtaining and evaluating and communicating information. And so there's got to be multiple science practices that students are explicitly supported in engaging in. There should be a discovery element. It goes back to research. We do research, we don't already know the answers. And so for our cure, students didn't know what levels of nitrate or nitrite they were going to find in their films. Neither did we. Nobody did. That was the whole point, is sampling different locations in the environment to look at what's in the films that form in those locations. The work should be broadly relevant or important, just like any other kind of research that we do. And in this case, indoor films is a growing area of research since it has direct impact on human lives. Every surface in your environment is coated with a film. And so understanding what's in them is important. Collaboration is another aspect. And so in our design, students had their individual samples that they picked where they were growing their film, but they worked with a partner as they developed their procedures and analyzed their results. And then iteration, so that opportunity for feedback and revision for data collection and procedural components. So these are always going to be multi-week experiences because, again, in authentic research, you don't always design everything perfect the first time. That as you get results, you optimize and figure things out. So Ellis and Kim will talk more about how their programs where students have these opportunities. But as you think about your own design, these are the things that you want to kind of double check and make sure you've baked that into the design. Next slide. And so I was going to do this at the end, and we decided at the beginning. So as you think about, so as you're listening to the rest of the examples of cures and thinking about your own program, you want to think about planning for publication, and it requires similar planning as in designing the experience for students. And there's 2 types of publications you can do with cures. There's educational publications, so focus on the curriculum. There's a few in JChem Ed in chemistry. So for that, you've got to be able to demonstrate the pedagogical content and educational relevance and insight that demonstrate a positive impact on teaching and learning. And for this, it can't just be cures improve student outcomes, because we know that work has been done. And so you're looking at what are you doing that's a resource that doesn't already exist. And so that's showing a connection to teaching and learning within the context of curricular or coursework, and then be supported by up-to-date citations. A lot of the cures that have been published, it's sort of here's the laboratory experience and the setup that people could modify to actually turn that into a laboratory. And then the other is research publications, where the focus is on the research itself. And for those would get published in analytical chemistry journals or wherever you publish the rest of your research. Those are not appropriate for education journals. Next slide. So if you're thinking you want to do an educational publication, one of the things that will be required is that evidence of classroom use. So you've actually implemented it in the classroom, typically with more than one iteration of use with students. If you're working with a couple of hundred students, we might say, okay, one time through would be okay. If, like most of us, you're working with 4 or 12 students, you're going to want a couple of times through to make sure it works well. You have to have clearly articulated learning objectives, which I would say you should have anyway, and if evidence that objectives were met. So if you've said this experience should teach these students these skills, then you need to have assessments that let you determine if students were successful in doing that. And that's that alignment. You need to be thinking about resources or insights for other instructors. And so what's going to go into your supporting information that allows duplication, that other people can take pieces or do it. The other thing that comes in the pre-planning, it does require IRB or explicit permission from students if you're going to use examples of student work. You can do the generalities that 80% of the students did fine on this assessment piece without those. But if you're going to put any examples, you either have to have a written statement of permission from the student or you have to have IRB. So just kind of keep that in mind. Those are much harder to do after the fact. And then next slide, research publications. It's the same rules as any other research publication. You have to demonstrate novelty and do the methods. The big thing with a cure is that you need to be very clear about authorship for students. That what are they doing that's going to earn authorship on anything that comes up. If you're putting in your educational paper, the students should not be authors. And I have actually handled a few of those where we made them take the authors off because it wasn't about their results. It was about how I designed this curriculum, and the students did not help design the curriculum. They implemented it and were subjects, not authors. For the research piece, if they're designing experiments, collecting and analyzing data, then they are the ones generating the information for that publication. And so that's a check journal requirements. Are there limits on the number of authors? What are the expectations for authorship? If you've designed it where they really are partners in a research project, it is appropriate for them to be authors. If you've got very many folks, I've seen this with some of my own work, and much more commonly is that journals want a list of who did what. And you get over a certain number and more are requiring you to kind of list each author. And then there's sort of, here's all the things that would warrant authorship, and you have to indicate who's done that. So thinking ahead of time about what that would look like and that everybody's on the same page in terms of expectations will be important to do that summary. And I would say that's probably a best practice if you are submitting something with 15, 20 authors, is just put that in when you submit as additional supporting information for the editor. And at that, I'll let the other two talk about the exciting work that they're doing in cures, and I can certainly answer questions at the end.
Thank you, Dr. Cole. Now, Dr. Bell.
Great presentation, Renee. Very important stuff. Following up just on what you said at the end, I'd like to put a plug in before I start going through our stuff for an organization called CureNet. And you can Google CureNet and find it. Erin Dolan, who in the life sciences is one of the big movers and shakers in cures, has created a wonderful organization, a wonderful site. And if you're thinking of developing a cure of your own, I strongly recommend that you go to that site and follow the guidelines, which, you know, Renée sort of hinted at a lot of them, but there's some really practical things in that. Every time I develop a new cure, I go back to it and work through their worksheets and things, and it's incredibly useful. So big plug for Erin Dolan's CureNet website. So we developed, our group with NSF funding developed something that's called the malate dehydrogenase cures community. And it started with about a dozen people in different institutions and has now grown to somewhere approaching 100 faculty in institutions actually now all over the world, doing course-based undergraduate research on malate dehydrogenase. But from the pedagogical research that we did during that, and we've published a lot of that can be found on the website, we came to the realization that there were many components of research that really need to be included to give a truly authentic research experience to students. And those are listed here. Some of them overlap the things Renée talked about. A couple of them bring in a couple of new aspects. So, you know, we're all research scientists and we've all published in research journals. And we know that our research has to be relevant. It has to represent appropriately the scientific background. It has to be hypothesis driven, and hypothesis development is an important part of research. And usually we have to present a proposal to get funding or resources. And students should go through all of those aspects. Obviously, they should go through designing and executing appropriate experiments. And that includes, as Renée pointed out, all types of collaboration. And it can be collaboration within an institution. It can be collaboration between institutions. And we've published some stuff on the pedagogical outcomes of collaboration that shows that students really get much better ownership of their project, and that leads to stronger student-centered outcomes when they collaborate with faculty and students from other institutions. It brings a sort of an immediacy to the work they're doing. Next, there's reproducibility. Thank you. You know, one of the questions that everybody that's ever taught a cure probably gets faced with is, did I get the right answer? And the response I always give is, 1, was it reproducible? 2, how much do you believe your data? 3, what's the data telling you? I don't know the answer, as Renée said, this is research. If I know the answer, it's not research. So, you know, that first question students usually ask is it the right answer? And I always throw it back on, how do you believe your data? What does it take to have believable data? Is it reproducible? Then obviously there's data analysis and drawing evidence-based conclusions and communicating the results of the research. And this can be in terms of a paper or an oral presentation or a poster presentation. And usually we try to build in elements of each of those. The thing that's not usually put on a list of things that make for authentic research are the things on the left-hand side here. Peer review. If any of you have ever managed to get a paper published or a grant funded without going through peer review, please let me know. Because I'd love for people to just say, yes, I'll publish a paper and yeah, here's money, without having to go through peer review. Actually, I wouldn't, because peer review is possibly the most important part of the scientific process. If we don't put as scientists, if we don't and aren't prepared to put our ideas out there and listen to others' opinions about them, we're not really doing science in its fullest. So we build peer review into our cures, and there are various stages. So for instance, when they do their proposal, it can be peer reviewed, and we do a mock NSF or NIH style panel discussion of people's proposals. We do peer review of data presentations explaining how you did the experiment, what the data, what you measured, what it means, etc. And of course, the final presentation is an obvious place for peer review. And peer review lets you build revision into the process. And students love to get feedback and be able to revise before they get graded on these things. So those sort of 9 attributes of research we incorporate into all of our cures. So if I can go to the next slide quickly, please. And so we've developed over the years a bunch of resources, including templates and rubrics for all of the things that were on that previous slide. And I'm just going to quickly point to that. Those are all links you can go to and whatever. So next slide, please, Renée. So one of the things we found to be a challenge in teaching research type topics, we've solved with this thing that we've called the totals approach to science. And students in general labs get given a rubric, a protocol, and they just follow it rotely, and they get the expected answer. That's not what science is. And so we try to bring this, as I say, totals approach. And what totals stands for is try something, observe what happens, think, and that's the thing students hate to do usually when they start this. They hate to think about, did I do this, or what was I expecting, whatever, but think, adjust and try again. And then the LS at the end of totals stands, if you go through this process, you end up leaving lab smiling and satisfied because you've learned how to do something properly and adjust on the fly. And we found we can adapt this to all sorts of things in our cues that range from sort of organic chemistry type things to analytical chemistry to biochemistry, etc. But that acronym appeals to students. Okay, so if we can have the next slide, please. And the hardest thing we've found over the years is to help students develop their own hypothesis. Too many people, and this came up at the American Society of Biochemistry and Molecular Biology meeting this year, too many faculty think they're teaching a cure because they're telling the students what the hypothesis is and what experiments to do, and then they let the students do the research. That's not a true cure. Students should be developing their own hypotheses. And we've developed this thing called the rule of threes approach to hypothesis development and proposal writing. And I'll illustrate it just quickly here. The first is the 3 somethings, and with a protein, all proteins have structure, they all interact with something, and they all do something. And so the 3 somethings are what part of the molecule are you interested in, what do you think it does, and what do you think the result of that interaction is. So, protein has structure, it interacts with something, and that leads to something, the 3 somethings. And then to convert that into a proposal, students have to ask themselves the 3 questions. And so they've got their basic hypothesis, and then the 3 questions are, what do I need to measure? What will I change to perturb the situation to cause the effect? And how will I know whether or not it had a significant effect and what it does to my hypothesis. And we've got worksheets that we give the students for the three somethings, worksheets for the three questions, and that really helps them take ownership of developing their own hypothesis. And then I think I've got one more slide. Quickly, nope, that was it. So.
Thank you very much, Dr. Bell. Now the word is going to go to Dr. Frederick, which I forgot to mention at the beginning, is a recipient of the Giddings Award for Excellence in Education. So Dr. Frederick, the floor is yours.
Thanks. So on this panel, I am the analytical chemist. So I'm going to acknowledge that we have some challenges when we think about developing cures for analytical chemistry. Those are primarily limited access to instrumentation, right? When we do research, oftentimes we use higher end pieces of instrumentation, of which we have for teaching purposes, maybe 1 or 2. If we have a core facility, maybe there's 3 or 4. But if you're teaching a lab section of 16, or if you're like at my institution where I'm teaching a lab section of 12, but we have at most 2 of any instrument and sometimes only 1, that limited access to the kind of instrumentation I use for my research means that it's challenging sometimes to translate a particular faculty member's research into an authentic research experience in the classroom. Going back. Measurements can also take a long time if you want to give students real chances for iteration. Auto samplers can happen, but if a run on the HPLC takes 20-30 minutes, the opportunity to look at that data and then make a real-time iterative decision is challenging. One thing that I have found, so I have been searching around for a course-based undergraduate research experience that I could do in my analytical chemistry class. And I found that method development as a process actually makes an excellent platform for a cure. So the figures of merit that you would do, accuracy, precision, selectivity, sensitivity, make excellent benchmarks for designing a cure. You can have multiple teams working in parallel. So working on optimizing different reagents or working on assessing different figures of merit. And students in the version of the cure that I developed before what I'm going to really focus on today, I had students working on method development for different ones of my colleagues. So colleagues in neuroscience and human physiology. And because they were working on different methods for my colleagues, they would be working on different instruments. So I will say that was it was very fruitful. We published, but it was challenging. I felt like I was doing teaching on crack as I was trying to shift between different people's projects that were very different. So after about 5 years of that, even though it was a great experience, and my colleagues in other departments thought we had superpowers because we were analytical chemists that could do method development, I went ahead and abandoned that for what I'm going to talk about today. So if you could go to the next slide, Susanna. So when we talk about cures, there's sort of 2 general categories. One is sort of individual, developed by an individual faculty member, and another is a networked cure where, and that's what Ellis is engaged in the malate dehydrogenase community. And that's what I'm going to talk about today with the microcure. So this is groups of faculty working at different institutions with their students and helping share, troubleshoot, develop resources, et cetera, together. And I should tell you that I'm going to show you the names of all of my collaborators in a couple of slides, but I want to do a special shout out to Rebecca Hunter, who is here. She is one of the microcure PIs. And you probably will have a lot of questions about this. And so I'm going to just tell you now, we're in the 2nd year of this grant. So we'll have a lot more answers in about another year, but we're going to go ahead and welcome folks to plan ahead if you're interested in adopting this upcoming in summer of 2025. So the way the microcure is focused is what we're doing is we're adapting solution phase assays into a micropad or a micro paper analytical device configuration. So simply what that looks like And this, I've simplified a lot of stuff into this slide, so it looks super simple, and in some ways it is simple, but there's a lot more to it. I feel like I just need to say that. So essentially what you're going to do is you're going to deposit reagents onto paper. Then you're going to put on your sample of interest, and we're looking for a colorimetric change. And in that colorimetric change, we're going to capture using cell phone imaging. And then we process the images using either a color detecting app or ImageJ and quantify that color change. And then we can establish a calibration curve like many other calibration curves, and establish figures of merit in that way. The strength of this is that really the equipment that you need is very minimal. Almost all your students have a cell phone, and you can go ahead, and this technology has been published in many analytical journals. So this is easy to find a mainstream analytical outlet. So if you could go to the next slide. So the overall structure of our MicroCure, and so Micro came out of the Micro project, if any of you are familiar with it, which was a COVID, initiated during COVID as a way to develop inquiry-based labs to students. So this is the Cure offshoot of that, just to explain where the Micro comes from. So the overall structure is that students with faculty consultation identify an assay from the literature that they want to adapt. That's really nice in that it allows them to personalize what they're interested in. If they're interested in the environmental applications of analytical chemistry, they can look for an environmentally related assay. If they're interested in food, they can look for a food-related assay. And as we know, there's colorimetric assays to analyze lots of things that have not been adapted into micropads. So you can even use chemistry from the 1920s, as long as it hasn't already been adapted into a micropad, it's fair game. So there's lots and lots of things to choose from. We have a number of exercises that we've developed that take students through this process, but that's the first step. And then students develop an overall process to optimize their reagents, optimize their standards, their approach to standards, and then determine the figures of merit. And what we ask them generally to focus on is accuracy, precision, linear range, sensitivity, selectivity, and detection limit. Students interact with faculty or a TA weekly before lab. They're responsible for analyzing their data, making sense of what the data says, and then proposing. And again, we have ways to train them and support them in that activity. But that's one of the things that they need to do is interact with the faculty either synchronously or asynchronously before coming to lab each week. And then typically the MicroCure lasts for between 4 and 6 weeks. I think there's one person who does 7, but I think the shortest so far is 4. And then it ends with a presentation, and that looks different. In our initial group, we have been doing cross-institutional poster sessions. So Rebecca was teaching this semester, as was I, and our students engaged in a poster session with each other. But other folks have had students write up lab reports or SOPs, some type of summative exercise that wraps things up. Next slide. So we chose collectively to work on developing experimental design skills, group work skills, evaluation of data, argumentation from data, and presentation. And for the ones that are marked in asterisks, we use the Ellipse rubrics that Renée and her colleagues developed, and they have been fantastically helpful. So I just put in a plug for Ellipse. If you haven't currently used those, they're designed to be taken in parts and chunks, and they have feedback. So there's really nice places to indicate where you would suggest your students improve. So highly recommend Ellipse. Oh, and sorry, I should also mention one of the things that I did was ask my students to reflect on the nonlinear process of research. Sort of what did they end up doing? And this is one of the figures that one of my groups on the right there that shows like, okay, well, we did this thing, but then we had to loop back. And this is actually a fairly simplified version of what they actually had in terms of the complexity of their research process. All right, next slide. So what I would say is the strengths of this particular project are that it's safe, right? If you're trying to give students a high degree of autonomy, you would like it to be in a situation where there's not a lot that they could do to hurt themselves. The nice thing about, especially, for example, if you were at a large university working with multi-sections, maybe run by TAs, because micropads use microliter quantities of chemicals, and often at low concentrations, the exposure risk that students have to even things that would be moderately dangerous at high quantity is minimal. So I tell my students, I'm like, you probably could eat the micropads. I would not suggest it, but you probably could, because it's a fairly safe technology. Because we're using cell phones, the amount of instrumentation and technological cost that's involved is very minimal. The measurements are rapid. From production of the chip to detection is often within 15 minutes. So that allows them to think of an experiment, or hopefully they've done that before they come to lab, conduct an experiment, analyze the data, make sense of the data, and then go on to the next iteration within a single lab period, which is a really powerful experience for them. Like, oh, that's what that means. Okay, I'm going to try that again, but slightly different or under different conditions. It's authentic. We need more micropads. Micropads are ways for people in non-lab settings to access chemical information. So these are publishable. I have a paper out for review right now with analytical methods on one of the methods we developed, not this past semester, but the year before. And the nice thing about this project is it's scalable. If you have 8 students that you're teaching, you can do it with 8. If you have 64 that you're teaching, you can do it with 64. Want to think about group size, of course, so that you can manage your own cognitive load and that of your TAs. But because of the relatively inexpensive cost of the technology and the relative safety and small amounts of chemical exposure, you can do this pretty easily with lots of students. All right, next slide. So if you're interested in getting involved, these are the 4 PIs on our NSF grant. Myself, Rebecca, who's here and can also answer questions, and Maury Howard at Virginia Wesleyan, and Daniel Scott at Centre College. And we'll be looking for collaborators in the summer of 2025. And what we're doing this summer is developing all those resources to support you in implementing this. So if you indicate interest now, we'll be able to get an idea of what the materials you'd want to have in order to implement. And I think that's my part.
Thank you very much. And thank you to all our presenters. We have also added a couple of slides with additional free resources, such as CureNet that Dr. Bell already mentioned. And then you also have some open access articles and additional science networks that you can join. And again, this PowerPoint is going to be shared with you all, so you can click on these links easily and get to the resources that you need. With this, we are almost at the end of this session, and we are going to be proceeding with questions. I think for that, I will be getting out of this view, the PowerPoint view, so we can see each other. But before I do that, I just want to say special thanks also to Dr. Betty Galareta and Dr. Michelle Kovařík for helping us to put this session together. And I want to make sure that you're all aware the ACS Division of Analytical Chemistry Education Committee organizes many of these panels throughout the academic year. And if you have ideas for topics you would like to see covered, either something that should be addressed only just for students, for faculty and students together, or just faculty, then please do email Dr. Kovařík, and she will be more than happy to then bring these ideas to the education committee. And thank you very much to our presenters and to all of you for joining us today. And I'm going to stop share and put us all in the view where we can see each other. Okay, thank you. Michelle, do we have questions in the chat?
There aren't any in the chat, but maybe people can put some in or raise your hand if you'd like to ask something.
Oh, Renée, yeah, Dr. Robinson.
All right, thank you all. This is super interesting, and I'm sure a lot of coordination behind the scenes to make it all work. I had a question about the rubrics and the assessment. So generally when you get to the, or I guess as you're going throughout, how are you giving feedback to students to let them know they're on the right track? And then by the end of the semester, how are their final grades assessed? And do the students feel like they're okay with the grade they received because they had clear communication about what the expectation looked like throughout the course of the semester or the 4 to 6 weeks?
Dr. Bell.
So that's a great question. I'm glad you asked it, because grading is obviously always at the top of students' minds. And I'm going to put a plug in for an ACS book that will be coming out, actually probably around the time of the fall meeting. Betsy Martinez-Vaz is editor of a book on cures and all aspects of cures. And one of the chapters is on grading and the various ways that you can incorporate grading into a cure. So look out for that over the summer. It'll be coming out. But grading, the syllabus that you give the students at the start of the semester, I try to be very explicit about what the students are going to be graded on. And it's really, I mentioned peer review and things like that, and revision of, there are various products that they're going to produce, like the proposal, like, a PowerPoint or whatever. And we try to base all the grading on that and give the students a chance to revise and learn from the revisions. And we use a sort of a give back clause in the rubric, saying that they'll get an initial grade, and then if they revise, they can get back 75% of what they missed if they missed anything, things like that. And that gives the students obviously an incentive to go through the peer review comments and think about what they should have said or how they could have made their presentation or product better. We try to avoid grading on the actual experimental outcomes, but how they think about them, how they present them, get away from that, is it the right answer issue. Does that help, Renee?
I was just going to add, one thing that's really important is if you're going to do a cure for at least part of your lab curriculum, that you think about how you're scaffolding the earlier labs. So for example, if you do 10 weeks of, or sorry, 10 weeks is a normal semester. If you do 5 weeks of cookbook labs where you're, just having them type in their answer into a spreadsheet and grading them on accuracy. And then you're like, and here's the cure. That transition can be really jarring. So in the microcure, we're working with Kelly Ellis at St. Mary's College of Maryland to develop some backward design resources to help people think about, well, how do you set them up for success in the cure? She's got a great article. I'll put her name into the chat. She's got a great article on backward design of lab curricula. With some really nice resources, that worksheets that you can work through that'll take you through that exercise. And then just echoing something that Ellis said, students get rubrics ahead of time they get a full point distribution, but they also get rubrics ahead of time that say, okay, for this assignment, these are the things I'm going to look for. And they get feedback on that in week 1. And then we talk about it. And then they come back, for example, for their weekly update. They get to try again, and they get to try again for each week. So they know how we're going to grade them. It's going to be based on process, not actual experimental outcome. And they get it before we start and have multiple opportunities to reach higher and higher and higher.
Yeah, I would say this is also, and I see Amber's got her hand up too. This is also a good opportunity for folks who are exploring ungrading or proficiency grading. We've done that in some of our upper level classes where it's all about that iterative. Students need to demonstrate their proficiency at the different skills. And as long as you've got really solid rubrics, then that lends itself really well for that. But they do need feedback along the way. You can't wait and just grade a final project.
Thank you all.
Dr. Amber Hopp.
Thanks so much. I'm curious, you just mentioned it a little bit, Kim, but I'm curious kind of the setup and how you're thinking about what do you give to the students before the cure? You know, whether you're doing a semester-long project where they've maybe had a whole semester of instrumental beforehand versus like putting it into a semester of instrumental. You know, I'm trying to think about the balancing of the skills that they need to build, because sometimes they're pretty terrible, especially these COVID kids coming into lab, their hands are not what they used to be. And so trying to build their skill up so that we can believe what they're getting out of the cure. So I was just wondering if you could talk a little bit, any of the three of you, kind of about how do you balance the cookbook labs where they're gaining skill, particularly rather than content, more skill, with the cure piece and the more independent piece.
Right, I think that's a great question as well, Amber. And when you're first thinking about putting together a cure, when I first started doing it here at the University of San Diego, when I moved here, the department had certain requirements for certain labs, you know, certain skills that students had to come out of the lab so that they were ready for the next lab in the sequence, etc., etc. And at first, there was some reluctance to people doing cure type labs because it wasn't going to check all the boxes on the departmentally agreed list of skills and whatever. But I went out of my way when I first started doing it in general chemistry to show my colleagues how each of the skills was being met during the cure. And so, for instance, I teach a full semester cure, and they learn techniques, and I make sure the techniques they learn are ones that are going to come up in the cure part. And so we take the position, as research often does, is you need to be able to reproduce something that's already known and build from that. And so they learn some of the techniques in the first few weeks that they're going to use later. Now, some of my colleagues in the GChem sequence, and we've got one person that teaches an analytical chemistry one person teaches a material science one, but they do it with the sort of the modular version where they teach them the cookbook labs for the first 6 weeks, and then the last 6 weeks become the cure. Both, and we've done pedagogical research on that, and those modular labs where they only do half a semester actually have many of the same benefits as the full semester cures. And so it's really a matter of working with the department and making sure that the cure component hits goals that need to be hit. And, you know, we're all creative faculty folks. We can usually work out how to you know, sell something that it's going to hit a certain set of benchmarks. So, for instance, you know, they have to learn some spectroscopy, and they have to do standard curves and things like that. Almost any research project can be incorporating spectroscopy and standard curves. You know, the beautiful one with the phones. I love that. I'm going to work out how to adapt that one to what we're doing. But, you know, it's a matter of sitting down and thinking about what the department edicts are for what needs to be covered.
So Amber, to address that from an analytical perspective. I happen to know Amber and know that Amber only has 1 semester of analytical to teach as I do. So figuring out how to balance getting exposure to HPLC and AA and the sort of standard methods, along with then I dedicate 6 weeks of my 12. I generally am mean and ask them to come to lab that first week. So I get 13 weeks. They dedicate half of that to the cure. And so each of those labs leading up, actually none of my labs are cookbook, but all of them that have multiple objectives. For example, my HPLC lab also does pre-treatment with solid phase extraction and a tiny bit of method optimization around chromatography separations. My GC lab also does solid liquid extraction and looks at internal standards and how do you treat internal standards when you have an extraction step. So I've just really thought sort of intentionally about how do I get them the exposures that I need. And then my own reconciling with this is that it is more important for them to have a lot of time to develop transferable skills around troubleshooting, method optimization, method development, than it is for them to have a lab where I'm like, okay, now vary the percent organic of this mobile phase. What does that mean? So that's sort of how I reconcile it. Is it right? I don't know. I don't know that there is a right answer. Talking my colleagues into a second semester of analytical is probably the right answer. But that's how I do it, is that I think, how many students are really going to go into analytical, let's say at the graduate level, maybe 2 or 3 in a year. How many of them are going to have to optimize a method and validate it? Oh, probably a lot more than that. And then the HPLC they might use for me is probably different than the HPLC that they're going to use in an industrial context. So how much do they need to know how to operate my HPLC versus just having been exposed to it?
Thank you both. That was helpful.
And Allison had a question in the chat for me, so I'll just really quickly respond to that. So there are certain things that work well with micro pads and certain things that work less well and certain things that I know less well. So I will work with them. So for example, if they identify, like we did a caffeine assay this year, and a lot of the colorimetric reagents had like chloroform extractions. Well, chloroform extractions don't work well with hydrophobic barriers and microfluidic devices. So we kept working until we found an assay that was for the analyte they were interested in that would be adaptable. So once they sort of narrow things down based on their own interest, I do work with them iteratively to identify an assay that would work.
The other question that was a little further up that I think would be appropriate for you is, how do you deal with the logistics for ordering supplies if students get some agency in designing their experiments?
Mm-hmm. So I have a money limit for them. So, you'd be surprised if as long as you have a chemical inventory that spans all of your colleagues, you'd be surprised. There's lots of things there that are already available, especially when you might only need, a half a gram at most for the entire project. But if they need things that are new, I put a money limit on them. And because our end goal is an assay that's going to work for a non-technical audience, if it's going to use something really, really nasty, then I'm like, okay, that's not going to be something that we're going to do, because that's not appropriate for the end use of that assay.
Yeah, and I'll just add in this about prep for lab and things like that. Because usually GChem labs have, they're all the same. One person preps them all, one person sets them up. And Cure labs just don't lend themselves to that. My GChem lab is on drug design, and I've got 18 students doing 18 different things, needing 18 different types of reagents or experiments at any given point in time. And so I actually get the students to do the prep work. They learn to make up their own reagents. They learn to think about how much of a reagent they need. And they've actually, the feedback I've gotten from the students is that that's been the most valuable part of the course often, is that instead of being handed something and just told put X into Y, they have to think about it. They have to design how much they have to weigh out and make up. And, we go through the department's inventory and find all the reagents, and then the students have to make them up and prep them. And that's become a very valuable part of and faculty that get those students in their research labs respond, boy, these kids know how to do stuff. And so that's a benefit.
And I will say that's the same for me. I actually, except for getting, I have a TA who I then have prepare the chips in terms of just physically printing them, and then we have to bake them as well.
Right.
All the solution prep is done by the students. And so it's like, oh, great.
Yeah.
Time to go to lab. Woo! And there's no prep ahead of time.
Yeah.
Michelle, did we address all the questions in the chat?
I think so. There was sort of, Kim addressed most of Allison's question, but I did think there was sort of an interesting second half of that of, do you find students repeating prior year's work, or do you show them what past students have done? I'm kind of curious, or do you try not to? So I wondered if you all could speak to that a little bit.
So for me, if students haven't, so I'll be totally honest, here's a little moment of transparency. Generally what happens is that students will start a project, and then my research lab will pick it up and finish it off. Because they can't get it, they can't get all the data for publication. And often, right, they need to repeat it and get inter and intra-day reproducibility, all that stuff. So they generally can't finish in the 6 weeks that they have. So if my students don't have time to pick it up, and I will say that sometimes I'll have groups that just really need a helping hand. And if we have an assay that hasn't been finished, then what I'll say is, hey, your job is to kind of finish this off. So I'll give them sort of where the last group started, and then they have to take it further. And there's enough, I always make sure there's a plenty left to do on that assay when they pick it up. But as far as like repeating other people's stuff, right, this is research. So if it's an assay we did before and we've published it, doing that again isn't research. So so I don't have that problem in terms of replication, because it's just not an option for them.
Yeah, but I'll add to that, because oftentimes what happens in even my first year GChem cure, a student will get to a certain point during the cure and then go work in the lab and get it to a point where they can submit an abstract for a meeting that gets published. And so when we do the literature research sort of background, we make an emphasis on looking not just at full papers, but also published abstracts, so that students can actually discover what has been done, either by our students or other students on topics. So that seems to work well. But as Kim said, there's an aspect of reproducing.
If you think we have time, there's one more question in the chat. It's how do you cope with failed experiments? So I guess I'm curious about this too. If you've ever had a group that had sort of almost no data, then what do they write about, or what do they produce, or how do you work with them on that?
So for me, when my students do their proposal, they have to have at least 2 different methods that they have for the analyte they're interested in studying. So I had a group this year, the first week. So right, they're developing a solution phase assay. They're starting with a solution phase assay, and even the conditions that were in the paper exactly when they tried it on pad didn't work. I had them do it in small quantity in like microcentrifuge tubes, didn't work. I was like, okay, guess what? You have a backup and you have it ready, and we're ready to go. And as long as they can get some color change, then they can always get enough. So what I will say is that complete failure absolutely happens. But I've learned the hard way. The other thing that I do is sometimes I'll have previous years, or I'll have an article that I was like, oh, this would be a cool micropad. So sometimes I'll have that like pocket, like, well, that didn't work, but here, why don't you try this? because it does happen.
Yeah, and I think it's important to students to realize that something not working is actually a result if it's reproducible. That, you know, this approach isn't going to work, and we've learned something. Not let them think they've failed. They've progressed our understanding of how to do something.
Yeah, and I think that's important. It's that science is about process and exactly that it's not always that it has to work. It's the, okay, this didn't work. What might have been the factors for why this was an appropriate method, or why this didn't go as planned. And if you can get students to do the troubleshooting piece, I think long term that's more valuable.
Right.
Thank you very much. If there are no further questions, then we conclude the session. And thank you again to our panelists. This was a great session. Thank you to all for attending and giving us even greater questions to move forward. And we hope you will be implementing Cures into your curriculum. And we will be emailing the recording as well as the PowerPoint, so you will have all the resources that were shared today. And with this, I conclude the session. Thank you very much, everyone.
Thank you.